So we are talking about the photosystems of the light-dependent reactions, and remember that there's two different photosystems. And there's actually two different ways that the electrons can move through these photosystems. So we're going to learn about both of them. The first one is linear, which is when they start in one place and end in another place. And then there's cyclical, which is when they start in one place and they come back to that same place. So we go from a line to a big cycle. So linear electron flow is when we make ATP and NADPH. So we get two products from linear electron flow. It involves both photosystems. So those are two important things to remember. It involves both photosystems, and we get ATP and NADPH. So what happens is a photon hits a pigment, in this case usually chlorophyll A, hits chlorophyll A, and it gets excited. So it gets excited, and the electrons get excited, and when the electrons fall back down to their ground energy state, the pigment next to it gets excited. So it basically bounces around. The energy bounces from pigment molecule to pigment molecule all around the photosystem. Remember, we're at photosystem 2. That's where we start off with. And then finally, it gets to P680, which is a special pair of chlorophyll A molecules. So we're in the whole photosystem, and it's bouncing around from all the different chlorophyll molecules in there, and then it lands at P680, which is the special pair of chlorophyll molecules. And from there, the electrons get excited at the chlorophyll molecules at P680, and then they give them to the primary electron acceptor. So now they have arrived at the primary electron acceptor right here. So we can see the photon comes in. Remember, photon is basically a piece of light, which sounds silly because we talked about how light is actually waves, but in this case, it's a discrete particle of light. So we can talk about it as in it's one thing. So a photon hits P680, or I'm sorry, a photon hits a chlorophyll molecule in the photosystem. Then it excites its electrons, and basically the energy gets passed along from chlorophyll molecule to chlorophyll molecule. So finally it arrives at this special pair of chlorophyll A molecules right here in P680. This right here specifically is called P680. Then P680 donates these electrons to the primary electron acceptor. So step 1 is the photon hitting and getting passed around, and step 2 is when the electrons are given to the primary electron acceptor. And so when P680 gives away its electrons, it becomes P680 plus because it got oxidized. It gave away electrons, so now it's more positive. So P680 is a very, very, very strong oxidizing agent, which means that it is very likely to oxidize another molecule, which means that it's going to take electrons from someplace else, which is why the electrons end up there. If it's an oxidizing agent, that means it's being reduced. If it's being reduced, it's gaining electrons. So P680 plus is very, very likely to end up with electrons, which is why when the electrons bounce around, they always end up at P680. It's the most electronegative. And so then what happens is there's water floating around, and the water is split by enzymes. So this is what we were talking about in class the other day. The water is split by the enzymes and becomes oxygen and hydrogen and electrons. And so the electrons are given back to P680 plus to replace the electrons that it just gave away. So now it goes back to being P680. So P680 gets the electrons from all these chlorophyll molecules. P680 gets the electrons and gives them away. Now there's kind of like, it doesn't have electrons, so it takes electrons from water. And then water becomes hydrogen and oxygen. And so oxygen is released as a byproduct of this reaction. So water gives its electrons to P680 because it doesn't have any because it just gave them away. And now the oxygen is what leaves from the water. And that's where we breathe out water. It's from photosystem 2. That's step 3, is water splitting and replacing the electrons that P680 just gave to the primary electron acceptor. So now these electrons have reached the primary electron acceptor, and they're going to fall down an electron transport chain from photosystem 2 to photosystem 1. So they're at the primary electron acceptor of photosystem 2, and they're going to fall down an electron transport chain until they reach photosystem 1. Now, the energy is released as these electrons are falling down. So they start off as really high energy, and they end up at really low energy, and so that energy has to go somewhere, right? Energy is neither created nor destroyed. It has to go somewhere, and so that energy is used to pump hydrogen ions across the thylakoid membrane. just like how the same energy from the electron transport chain was used to pump hydrogen ions across the cristae during cellular respiration. So the electrons falling down the electron transport chain between photosystem 2 and photosystem 1 release energy, and that energy is used to pass electrons, to pump protons across the thylakoid membrane. And when that happens, the protons then rush back in through ATP synthase, and we make ATP by exactly the same process that we made ATP during cellular respiration. So now we have some ATP being made after the electrons have passed through photosystem 2. When they're done with all of this, they arrive at photosystem 1. So, we're at photosystem 1. Now, again, a photon hits chlorophyll A in photosystem 1, excites an electron, gets passed around to P700, not P680, now we're at P700, and P700 is really electronegative, so it grabs the electrons and then it gives them away to the primary electron acceptor of photosystem 1. So, another photon hits, bounces energy around until it hits P700, then P700 gives away its electrons to the primary electron acceptor of photosystem 1. And so, how this is different because in photosystem 2, P680 got its electrons from water. In photosystem 1, P700 gets its electrons from the electron transport chain, from photosystem 1. So that's the difference between the two of them. Okay, so you don't need to worry about knowing what paradoxin means. But basically, these electrons are high energy. They pass down another electron transport chain, and this time NADPH is made. Not NADP, or I'm sorry, not NADH, but NADPH. So photosystem 2 makes, photosystem 1 makes NADPH. And so now this NADPH can go on to power the Calvin cycle. So this is linear electron flow. Basically, a photon shoots these electrons up to the top of photosystem 2. Then photosystem 2 falls down this mill, the electrons. This mill makes ATP, so from photosystem 2 we get out ATP. Now the electrons are back down to a lower energy state. They get excited by another photon, that they get shot up to the primary electron acceptor up here, and then they get used to make NADPH. That is linear electron flow. This right here is a great analogy for linear electron flow. This picture is on page 195 in your textbook. Then, we have something different called cyclic electron flow. In cyclic electron flow, we only use photosystem 1. So photosystem 2 and P680 are not being used. And this only produces ATP and not NADPH. And no oxygen is released because we don't need to split water again. We're just cycling the electrons. And the reason for this is because the Calvin cycle needs more ATP than NADPH. So we need to produce at sometimes more ATP from the light-dependent reactions than NADPH. So sometimes when that happens, we turn on cyclic electron flow, which only makes ATP. So what happens is the electrons in P700 get excited, they jump up to the primary electron acceptor, and instead of going forward to make NADPH, they go backwards and back down the electron transport chain, so ATP is made. And then they're back at P700, get excited, fall down the electron transport chain, back to P700, ATP is made. Get excited, go to the electron transport chain, fall down the electron transport chain, ATP is made, back to P700. So that's why it's cyclic, because it's just happening the same thing over and over and over again. So some organisms, such as purple sulfur bacteria, only have PS1 and not PS2. And they think that cyclic electron flow evolved before linear electron flow. So this came first. And then later, we evolved this pathway of a second photosystem. And cyclic electron flow can help protect a lot of cells from light-induced damage. Meaning too much energy causing damage to the plants. So then your textbook talks quickly about the comparison of chemiosmosis, so using an electron gradient or a proton gradient to make ATP between chloroplast and mitochondria. So this happens in both of them. Happens in cellular respiration and it happens in photosynthesis. So both chloroplasts and mitochondria generate ATP from chemiosmosis, but they use different sources of energy. So mitochondria transfers chemical energy from food to ATP. It gets glucose, completely oxidizes glucose, and then it makes NADH and FADH2 to later make ATP, whereas chloroplasts take the light energy to turn it into ATP, into chemical energy of ATP. So obviously, there's differences in the structure between chloroplasts and mitochondria. The structure is a little bit different, but basically the setup is the same. The whole function of it is the same. So in mitochondria, remember that protons are pumped into the intermembrane space, and then when they rush back through the cristae into the matrix, they make ATP. Whereas in chloroplasts, protons are pumped inside the thylakoid space. So here's a thylakoid, they're pumped inside here, and then they rush out back into the stroma to make ATP. So here we have it in both mitochondria and chloroplasts. See, it happens here on the thylakoid membrane, chloroplasts here on the cristae in mitochondria. So we pump H pluses via an electron transport chain into a small space, whether it be the intermembrane space on the mitochondria or the thylakoid space on a chloroplast. Then they rush back through ATP synthase to make ATP, and they're either rushing back into the matrix if it's happening during cellular respiration, or into the stroma if it's happening during photosynthesis. So basically what you need to know about the light-dependent reactions is that they make ATP and NADPH in order to power the Calvin cycle, which is the second step of photosynthesis.